All right. Hey, everyone. Welcome to our Sunday gathering in Scally Chucktonut's Legacy, working to educate, spread awareness. It's kind of what we do. And we have a really good show for you today. Very special guest, Cindy, who we'll get to in just a minute. We're going to learn about some harbor porpoises and some other work that Cindy's done. So we're really grateful to have you on. Let's go around the room real quick and check in. Deb Cruz, how are you? And we always start
Real
with Deb.
quick.
Take stuff all. Good to see you, Deb Cruz. Christina, how are you?
I'm good. I'm happy to be here. Happy to learn some stuff about the porpoises and talk about the J-pod last night.
Okay. Awesome. Brandy, how's it going? California, how are you?
Going good. We have nice weather, and it was awesome seeing J-pod last night. But I had it on. I wanted to put it on my TV inside, but my family would have probably been like, "What are you doing there in the middle of the show?" So, doing good.
It would have relaxed everyone. It was beautiful. It was a beautiful thing to watch.
That's what I think, because it was so
really, really great. Deborah Toro, how are you?
I am good, friends. Holding tight, anchoring in the love and light. So,
thanks for being here. And we'll talk a little bit about the love letters and the delivery coming up this week. From Deborah, thanks so much for doing that. Let's check in with Howard. And on this check-in, Howard, if you don't mind, what did you watch? Did you watch the webcam last night? Was that Center for Whale Research out there? What can you tell us?
Yeah, that was great. J-pod made this nice slow procession up the west side going north. And they went right by the lime kiln webcam. So I enjoyed it like everybody else, seeing all the babies. And it was just great to see them go by. And the Center for Whale Research got out there about 4 o'clock. And so there will be a complete report of all the individuals, but the baby J62 was seen, so looking real good. So that was great. That was a nice little surprise. I didn't see that coming. You know, there was J-pod right there off the west side, like the old times. But we also have at least 5 known gray whales that are out on the east side of Whidbey Island, mucking around for shrimp in the mud. And there's some transients. I don't think they're identified off Kingston, down in the Possession Triangle in Puget Sound, just south of Whidbey Island. So the waters are alive with whales.
That's so awesome. Thanks for, and you know, I was out sick this week, out meaning in bed, and so I wasn't on social media too much, but any indication if there's if we know the sex of J62 yet?
No.
Okay.
Haven't heard any sign of that yet, but maybe when the center gives their report in a few days, we'll know that.
That's what I was hoping for, because they were very active. They were very, very active. So hopefully they're able to get an undershot there. That would be great.
Yeah, I imagine.
How are you in Key West?
Good. We're experiencing, unfortunately, a little red tide right now, which is kind of unusual for us all the way down here. Typically, it occurs a little further up on the southwest edge of the mainland, but right now we're actually experiencing a red tide. So it's a bummer. It's all from releases of water from Lake Okeechobee. It's just nutrient rich, dead water. And we just need to do more for the Everglades and the water flow. Once again, a water flow issue, just like those dams.
Yeah. Does that start with the agriculture north of Lake Okeechobee?
Yes.
Yeah.
Big sugar.
Yep.
Big sugar has a lot to do with, how they appropriate the water.
Right.
And maybe that's something, Alma, we can do, get a show dedicated to that, because there's a lot that people can be doing and should be doing.
Absolutely.
unfortunate to hear that they're
We have
experiencing that.
a colleague in Florida that did some study on how the red tide up in Sarasota affected the bottlenose dolphins that were there and social structure and kind of what they did when the red tide was around, which was interesting.
It's interesting. Yeah, I'd like to learn more about that. Speaking of learning more, I'm going to ask everyone who's watching right now to click the share button on the bottom of their screen, whether you're on your computer or your phone. Share it to your page or to groups, because we're about to get some education, and you don't miss this. Alma, I'm going to let you do the honors of introducing Cindy today.
Cindy, I have some great experiences in life, and in several of them, Cindy was there. I've had the distinct pleasure of collecting underwater dolphin poo in the Bahamas with Cindy when she was working on her doctorate. And that was an amazing experience. And also we helped release a juvenile spotted dolphin together here off the coast of Key West. So we got to release him with his kind, which was just an amazing experience.
Yeah.
So Dr. Cindy Elliser, it's so nice to call you doctor now.
Thank you.
She's been researching marine mammals for 25 years now, authoring numerous scientific papers and presenting at international conferences. She's studied spotted and bottlenose dolphins in the Bahamas with Dr. Denise Herzing and the Wild Dolphin Project, and harbor porpoises and harbor seals in the Pacific Northwest. She's the founder and research director of Pacific Mammal Research. Her research focuses on long-term monitoring using photo identification and behavioral observations to learn about marine mammal, social, community, and population structure, and their behavioral ecology. Pacific Mammal Research is one of only a handful of organizations in the world utilizing photo ID with harbor porpoises. Dr. Elliser lives in Anacortes, Washington, with her husband, 2 kids, and numerous family pets.
There's too many to count.
Welcome.
Thank you.
Welcome to the show.
Thank you so much for having me. And I just want to say one quick thing about when we were collecting fecal samples in the Bahamas, us and many other graduate students and Alma when she was out, we called ourselves the femme fatales.
There were so many good poo jokes and puns. Femme fatales.
Yep.
That's awesome. Cindy, what you got for us today? I'm excited. I'm a sponge, and we've been super focused on the dams, which we should be, and we will continue to be. But it's kind of been our focus, and so I feel like personally, I'm excited to learn something today about the harbor porpoises and other work that you've done. So let's get to it. You tell me when you're ready for the presentation, or anything.
You go ahead and put it up now.
Yeah. Okay.
Yeah. We do research harbor porpoises and harbor seals here in the Salish Sea. I'm really going to be focusing on the porpoises. We just released our long-term 11-year study paper that much of the information that I'm going to talk to you about today came from that. I just want to side note, we also do photo ID on harbor seals. And so we look at their spotting patterns, and we can look at their site fidelity and behavior and stuff like that. So we've learned some other things about harbor seals, which maybe you can ask about later. But I just want to say that we do that, but I'm going to focus on the porpoises. They kind of take the front spotlight. So first off, I wanted to start with the differences between a porpoise and a dolphin, because a lot of people don't actually know. And a lot of times porpoises are kind of like left off to the side or ignored. Everybody thinks of whales and dolphins, but a cetacean is a whale, a dolphin, or a porpoise. So porpoises have, they are basically, let me see, the slides are not, oops, there we go. So the porpoises have the biggest difference that you'll see is their dorsal fin. So porpoises have a triangular dorsal fin, and dolphins have a more curved, or what we call falcate dorsal fin. And the dolphin species I have up there is a Pacific white-sided dolphin, which is the other small dolphin we have up here in the Salish Sea. So they have triangular versus curved dorsal fin. You can look at their face, and they don't have a beak or a rostrum like dolphins do. So if you think of like a bottlenose dolphin, they have that very distinct beak that comes out. So they don't have that. Porpoises have a more, most species have a beak or a rostrum, but then if you were ever lucky enough to look in their mouth, you'd see that porpoises have spade-shaped teeth and dolphins have cone-shaped teeth. So a little bit different there. And then socially, we know very little about harbor porpoise social structure, but generally they are seen in smaller groups and seem to be less social or very different type of social than the more gregarious and kind of hanging together a lot dolphin families and dolphin species. So that's the difference. So everybody now knows what a porpoise is. Yay. So harbor porpoises in the Salish Sea. So I'm going to tell you just a little bit of background about general harbor porpoises and then talk a little bit about what they are in the Salish Sea. So porpoises are the second smallest cetacean. The only thing smaller is the critically endangered vaquita in the Gulf of California. And so harbor porpoises are 5 to 5.5 feet, 150 pounds on average. So kind of a human size. And their nickname is puffing pig because they are very loud when they exhale. It sounds like a much larger animal. And then they're kind of short and squat like a pig. So that's a fun one. And they live anywhere from 8 to 24 years old, which basically means we don't know. And I'm going to tell you a little bit about what we found out about longevity a little bit later. But that's kind of the basics on harbor porpoises. So here in the Salish Sea, harbor porpoises are found all over the world in kind of colder waters. In the Salish Sea here, their history is that in the 50s and 60s, they were everywhere. But by the 90s, they were all but gone, especially in Puget Sound and the South Salish Sea. But they've recently come back. So there's anecdotal evidence starting in the early 2000s that they were returning in numbers. And so they finally went back out and did aerial surveys in 2016 and estimated that there are over 11,000 of them in the Salish Sea. And that's only the U.S. waters of the Salish Sea, because a lot of times research stops at the border, even though the animals don't. So we just don't know as much about what's happening on the Canadian side of that line. But so they're everywhere. And I like to tell people that you're more likely to see a harbor porpoise or a harbor seal than you are any other marine mammal when you go out on the water. So it's exciting that they're back. I like to talk about it because it's a good story, right? We have so much bad stories in the environment, but this is a good one. Like we've helped we don't really know why they disappeared, but it's likely due to, gill nets and pollution and habitat degradation and, doing stuff along the coasts. These guys spend most of their lives within a few miles from shore. So all those combination of factors, I think, helped for the porpoises to come back to their regular range. So that leads me to when I came out here in 2014. There was this place, Burroughs Pass, which is off Fidalgo Island. And if everybody's been up here, Washington Park is basically where our study site is. And then we know there were porpoises there from a previous work that had been done. And so it was a great place to, let's see if we can do something and research these guys, right? Again, porpoises have basically been ignored for the most part. And I, coming from the Wild Dolphin Project and doing research on the dolphins in the Bahamas, really interested in photo ID and social structure and looking at the long term and looking at through the lens of an individual. So we went to Burroughs Pass, sat on a rock starting in 2014. And we do know that the animals are there year round. And one thing that we have found very consistently is that they are less there in the summer. Not really exactly sure why, but they tend to just be in much lower numbers. They're still there, but many more days where we sit out and don't see anything. And so we have over 20, I think I can't see it on my screen now, but a lot of hours out there in the field. It's just sitting out on the rock. And that's one thing I want to emphasize. It really takes that. It takes just sitting out there and watching to be able to find out the information that we have over the years. And so that's the power of that kind of research. The other thing I want to focus on too is that it's not just me out there. I have a great team. We're all kind of part-time workers. We have to do other things to help us survive in the world to be able to do this work. But we spend a lot of time out in the field, and there's my staff. We have a lot of interns and a lot of volunteers that are crucial to us being able to complete this work and do what we've done. So we love everybody that supports us. It really helps us be able to do what we do. So when I came out here, I was like, is photo ID even possible with porpoises? Because no one had tried it. We do know from papers in the 70s that their pigmentation patterns are unique, but that was from stranded individuals, and nobody tried to do it with photos. Honestly, it probably couldn't have been done until digital photography, because only about maybe say 30% of the photos that we take are actually IDable. So it would be a lot of wasted film and a lot of wasted money at time. So with digital photography, it really allowed this to be able to do it. So it's really been attempted before, mainly because, I mean, they're really small. They're hard to photograph. When I first came out and said this is what I was doing, people honestly would laugh right away. They're like, sure you can. No, for real. They're hard to photograph. Sometimes all you see is the fluke print, which is that upper picture in the upper right. So it's they're just more difficult. So it hadn't really been attempted before, and I was like, well, let's try it. I'm a little crazy, so it's fine. So we did, and long story short, photo ID is possible with these animals. In the center there is a great illustration that Uko Gorder did for us, showing the different pigmentation patterns, and each individual between that dark and the light has individually identifiable pigmentation patterns there. And then sometimes they have scars and lesions, and that also, unfortunately for them, but helps us. And then their dorsal fin shape is really interesting. We can categorize them into kind of 5 different categories. And even within those, they're minute differences, but you can tell the shapes are really helpful in helping us identify the individuals. So we use multiple markers to be able to do that, because these guys are a little bit more difficult. But these are some of our very most cited individuals, particularly Pointer. She's our poster child. We've seen her over 125 times in the last 11 years. So she's one of our favorites. But we've published 4 different papers on doing photo ID, the most recent one being our 11-year synopsis of what we've done. So some information from our most recent paper, we've shown that harbor porpoises have up to 11 years of regular site fidelity. And we only know of one other place in the world that has this type of long-term data, and that's in the Netherlands, and they've shown up to 14 years of site fidelity for an individual. So most people are, again, not looking at these at the individual level. So we didn't know if animals stayed resident to an area or if they migrated out. And in some places it looks like they do, like in Europe, they do a lot of seasonal migrations, but in other places it looks like the animals are more resident to the area. So we have 164 different individuals. We've over half of those we've seen in 2 or more years. And like I said, we've had up to 125 sightings, or a little over that now, in the last 11 years for one individual, and up to 31 resightings in a year. So the animals are definitely hanging out in Burroughs Pass. We also have shown, I believe this is the first anywhere, that we have 3 generations of harbor porpoises that come to Burroughs Pass. So this is Piano, is the grandmother, and then her daughter is Piccolo. And then Piccolo showed up, and she had a calf we named her Piper. Actually, the internet helped us name her Piper. We had a little naming contest. And what's important to remember is that harbor porpoises can give birth, can be pregnant and lactating almost every year of their adult life. So they can have calves all the time. And both the grandmother, Piano, and Piccolo both had calves in 2023. So kind of fun. So we've also shown through this work natal philopatry. So that means basically that when you're born, you stay in the place where you live. So you can also call this like calf site fidelity. So Piccolo, we've seen 10 times over 8 years. Pizzicato is another one of Piano's calves, 31 sightings over 5 years. And Pharaoh, who is Pointer's calf, male calf, we've seen him 47 times over the last 5 years. And that picture down at the bottom, I'll show you a little bit more in a minute, but that's a mating attempt. They are very excited, and it's the one flashy thing they do. But the special thing that's special about that mating attempt is that is Pharaoh with his mother pointer. So at 5 years old, he decided to, I don't know if he knew he was trying to mate with his mom, but we documented that it happened, which is kind of cool. So a lot of the questions come up that porpoises have been said to be asocial, that they don't have any kind of social structure. But that's basically because they don't make whistles like dolphins do. So they thought they were quieter. They live in smaller groups. And so they must not have any social, they must not be social. But I think that is incorrect. It's certainly different than dolphin social structure, but we haven't been able to look enough into it by knowing the individuals yet to determine what kind of social structure they have. So they are often solitary. It's not unusual to see a porpoise by itself, but they can be in groups of like 1 to 3. But we have seen large aggregations in the Salish Sea. 100 plus is not uncommon. And for example, in 2022 alone, that we saw 160 of them. And this was work that was done along with the whale watching community and community scientists and other researchers to aggregate all the different sightings that we've had over the years. So it was a really neat collaboration to be able to determine that large aggregations actually happen a lot more frequently than we think. And those are about 20 or more individuals we call a large aggregation. So they're likely doing something social during that time. And we do see a lot of mating attempts and other kind of more social structure during those encounters. They do also like to play. They do wake ride. And unfortunately, the slides aren't, it's only showing the slide and not each picture in the slide. So this would be an actual wake riding picture where you can see it kind of going through. But it's interesting because they don't follow the boat like a dolphin does. They, someone just asked, how long do the aggregations tend to last? They can last anywhere from hours to months. So some aggregations in the South Sound, they've have been documented to go over 1 or 2 months. But other times it's just for a couple hours or a couple days. So when they're wake riding, the boats kind of go past or go through the pass, and then the porpoises go wee, and they wake ride a couple times, and then they go back to what they were doing. So they don't follow the boat like dolphins tend to do, but they do still do it. And this was, again, something that we didn't think that they did until we sat out and watched and said, well, actually, they do. And this was there's more pictures here on the how the porpoises eat. So they'll do surface chases where they come to the surface and dart across the water, and the water splashes up as they're chasing small fish. And so they normally eat small forage fish, less than 30 centimeters or about 12 inches, and things like hake and herring and anchovies and sand lance and things like that. But they have small bodies. They live in cold water. They have really high metabolism. So the unfortunate thing is these guys can't last as long without food like other cetaceans. Other cetaceans can last days to months without food. If a harbor porpoise already isn't doing well, it can starve in as little as 3 days. So they really need predictable food sources. So finding places where they can eat, and they have to forage a lot. Some studies have shown that they've maybe foraged 24/7, or at least close to that. So they have to get the food. And so this other picture with the gull taking the fish is the fact that gulls often take the fish from the porpoises when they are trying to eat. They like to steal it. So they don't. The porpoises don't always catch it. But sitting out there again while we were watching them, we've just discovered that they don't just eat small fish; they actually are taking salmon, and very large. And so remember that porpoises are 5 to 5.5 feet. Salmon can get up to 2 feet long. So they're eating something that's really, really big. And the first time I saw this picture was a colleague of ours that was out that first documented it. I was like, you can't eat that. What are you doing? Why are you doing that? But they catch it sideways, and then they have to orient it because they always eat things head first. So they swallow the fish whole and head first. And so the second question that came into my mind was, can you actually eat it? Or can you choke on that? Can the porpoises choke on that? And so we actually reached out to colleagues down in at the Golden Gate Bridge in San Francisco, and we asked if they had seen this large fish catching, and they had seen it with American shad instead of salmon. So we've documented it with salmon here. They were seeing it with American shad, which is an introduced species that was introduced about 50, 60 years ago into the Columbia. And so then we followed up that question, like, well, can you choke on it? Well, yeah, you know what? You can. Apparently, we looked into the stranding records, and I didn't put this picture on here because it's gory, and I didn't want to gore anybody out. But they can choke on it. There were 27 different cases of them choking on mostly American shad, and it's usually females. Over 92% were females, and over 87% of the fish were American shad. So, and so what's interesting is also that close to 87% of the individuals that had stranded and died with it in there were reproductively active females. So it's likely that if they could catch one fish rather than 200 small fish, maybe that was a better energetic investment. But the downside is you could possibly choke, and then you might take out, if you're pregnant, as well as the calf that you have that's with you. So it could be a bad thing if it happens too much. But so far, it doesn't seem to be too much of a problem. But occasionally, it does happen. So then remember that they do not prey share. They can't rip up food. So their teeth are too small. So they can't really do much. It's like smaller than a grain of rice is how small their teeth are. So they're really just for grabbing and then swallowing. So they don't, at least that we know of, they don't share food at all. Except when the eagles come in and take it too. So you can see the porpoise down at the bottom. This is a bald eagle. And so remember that the porpoises were holding the fish sideways in its mouth. It takes longer prey handling time at the surface to do that with the fish. And so we think that the eagles started being like, oh, well, that's interesting. They are very well-known kleptoparasites, which basically means they steal food, but usually it's from other birds. And so we documented that they steal from porpoises. We also have a picture of them stealing trying to steal from seals, doing the same thing, and even orcas. And I don't think I have that picture here. There was a bald eagle that went into a group of Biggs killer whales and grabbed some seal meat from them, which I thought was very ballsy of the eagle, personally. But he managed to come away with it. So that was something new that we documented as well. And then the last couple of things we'll talk about is mating. And I'm really sorry about this because this is the how the mating attempt happens, but it's only showing the last slide. But basically the male comes up on the left-hand side of the female 100% of the time. And it comes up and they leap like that. And it's like a second and that's it. It's the fact that they always come up 100% of the time on the left. We've documented that with colleagues around the world, everywhere we've looked. Which is a lot of places. And it's always that. And it's apparently the fact that their reproductive organs, the male and female, have evolved in a way that angle is the best way for them to do it. So if you've ever seen a porpoise leaping out of the water, like pretty far, they do leap for other reasons sometimes, but I would tend to guess that you probably saw a mating attempt. Very quick. And so we did just do a book chapter with colleagues on that. And then, of course, that leads to babies. Babies usually here in the Salish Sea come around August, September. So remember, our guys are kind of not around in the summer very much, and then they show up with their babies in August and September. So we think they go somewhere else to calve. We didn't know if they could calve every year or every other year because porpoises can do that depending on the population. Atlantic was thought to be every year, and the Pacific was thought to be every other year. But we've documented that they can have calves every year. We have an individual. We have a few individuals that have had successive calves in like 2 and 3 year spans. So basically, again, they can be lactating and pregnant every year of their adult life, which sounds exhausting. Having been pregnant, having kids, I would not want to do that. And what's very interesting, back to the longevity, is that 8 to 24 year time span. Most publications and the thought is that most animals are more like 8 to 10 years old, like that's the max. But we've documented at least 10 and 14-year-old individuals having calves. So Piano, that one that we've seen for 11 years, was an adult when we saw her. So she's at least 14 years old, if not more. And in Denmark and in Netherlands, they've also shown 12 and 13, 14-year-old females with calves. So it's likely that that number, that lower 8 to 10 number is too low for perhaps some populations that are otherwise healthy. So I like to end on babies because babies are adorable, and harbor porpoises are no exception. So I think that is, so that's my last slide there. And then I'm happy to ask any questions, and hopefully you learned a little bit about porpoises, and we'll talk some more about it.
Awesome. Thanks so much. We do have a question. Marcia is wanting to know what causes their decline.
So that's the one we don't know, mainly because nobody bothered to look at them, right? We have no data on porpoises really before the last like 15 or 20 years, very little, right? There's been some documentation of abundances and just certain locations, but no kind of long-term trends, especially back into the 40s and 50s. So there's no way for us to look at that and say, oh, that's why it was going down. But again, it's most likely a combination of factors with like gill nets were really bad, right? Gill nets are still the biggest killer of harbor porpoises in European waters. We don't have, luckily, gill nets here, except for very few. So removing of gill nets, their use, again, cleaning up the sound. So even though we still have a long way to go, we are better than we were in that, and also keeping, we're better about coastal development and those and habitat restoration and degradation and things like that. So those things probably help the waters get better enough for them to come back, but we can't pinpoint where they went or why they declined. Unfortunately. If somebody was looking at the porpoises back then, they might have known.
Okay. Sylvia is asking, could they have just migrated elsewhere, possibly?
Yeah. So what we know genetically from studies through NOAA is that the coastal animals, there's coastal porpoises on the outer coast, and then there's the Sailor Sea animals, and they are different stocks. They categorize things in different stocks. So genetically, those are separate. So it doesn't look like the harbor porpoises migrate out to the coast and back. Or like these guys basically, again, are resident. And from our study, it looks like we have a resident group that stays around Burroughs Pass. How far they go, we don't know because we need more photos from other places. We do know they at least go to Deception Pass because we have a match from somebody who was out on a boat. And somebody tagged us in it. And I was like, oh my gosh, that's a raindrop. So we do know they go that far. But other than that, we don't think that they really migrate here. They do think that the animals that came back into Puget Sound came from the San Juan Island area. So it's like they kind of moved up and still were not really around that much, but whoever was up there still came back down into Puget Sound when they retook the area.
I'm curious. Christina has a question too. I'm curious. I know Jared Towers had did this. There was an app or something where people can send in their photos, and it was like an identification process. I'm just wondering if that would be something that maybe, like some fundraising could get your organization, something like that, so people could send in, photos. And I'm just thinking of us sitting in the house, right, and Alma pulling out her big giant camera and capturing those harbor porpoises, just to try to assist. It's just a thought, that popped in.
Yeah, well, actually, we're talking with Jared right now. That fin wave is the one you're talking about with the big the killer whales. And so right now, the problem with the AI for harbor porpoises is that AI is great, but you still need to train it, and you have to have thousands and thousands of photographs. And so we have a lot of photographs, but not enough really great ones for as many individuals as you need to train that program. Having said that, we are working with them to get them our catalog and see if we can, if it can happen. but that would be ideal down the line. And we are looking into that as AI is getting better and as we get more photos and other people. But right now we do have our own sighting app. You can go to our website and click on the Get Involved section. And there's it's called Epic Collect 5. And then there's the Harbor Porpoise Project or Harbor Porpoise, Pac-Man Harbor Porpoise. And you can submit sightings of the individuals or sightings of harbor porpoises that you see in the Salish Sea. And then you'll have to email me the pictures because we haven't figured out how to have the photos be able to upload it in the app. But so we do have a few different community scientists that send pictures from around the Salish Sea. So we're starting that process, but we need more. So yeah, send photos if you have them.
This is cool. And I'm glad you mentioned the site because I did want to mention that to folks. It's really a cool site. I'm trying to get back to it outside of what I was trying to do here. Let me stop share and then I can start again. But the site definitely, and Deb Cruz just put it up there, mam.org. Google's going to say Pac-Man and you say no, Pac-Man.
Exactly. Well, that we thought, like when we made the name, I'm like, well, and it shortens to Pac-Man, we might get people accidentally, finding us because they're looking for Pac-Man.
No, that's really, really smart. But it's a very cool site. And so we can go into more details, but I don't want to take up I know there was a few other questions going on. Howard, did you have something?
One is a comment that maybe we can help generate citizen scientists sending in photos to you through our sightings list.
Absolutely.
So we can talk to our sightings team about that.
Yeah, that'd be fantastic. And we need more eyes on the water. There's only, 3 of us that are doing part-time work here with Sailing with Me. So we can't be everywhere. So yeah, the more eyes we have, the better. Even if it's and people go like, oh, I don't have a camera, I don't photograph. Still, we don't even know where the hot spots of where they are in the Salish Sea in a lot of places. So we need just where are they not? And so it's also important to note like, hey, I went out for however long and looked and didn't see them, right? So that negative data is always very important as well as positive data.
And my question was, I may have missed it, but do we know roughly the age of the first calf for the females?
Oh, when they first start having calves?
You're right.
Yeah. So because they do live shorter lives, most dolphin species live about 50 or more years. And even at the top range of 25 years, it's still only half that much. So they have to start having babies earlier. So they start having babies at age 3 or 4.
Okay.
So that's why we estimated when we saw Piano as an adult, she was at least 3, and then we've seen her for 11 years. So she's at least 14 years old, if not older.
Yeah. Okay, thanks.
Mm-hmm.
Christina, you had a question?
Well, actually, it kind of wasn't a question. It was just on the lines of what Marcia was talking about. And I just wanted to remind everyone that the harbor porpoises are on the 137 species list that rely on the Chinook salmon. So just another reason to breach the dams. That's all. I think you answered all my questions. I love your names. I just want to say that they're so cute. I love it. I love having names for them, and it makes sense that there's some like kind of like the transients out there and some resident. It's really cool. I love what you do. Thank you so much. I appreciate it.
Thank you. Yeah, no, there's always a debate in science whether do you number them or do you give them names? Some feel it's more anthropomorphizing if you give them names, but honestly, it's just easier to remember than like the big killer whales. When I learned their name, like the why they have that lettering and naming TA154 whatever. I was like, oh, that makes a lot of sense, but that's so hard to remember. Like TA1B256, it's like playing bingo. But so for me, it's easier just to have a name, and usually the names have some link to the markings that they have, so it makes it easier for us to identify them. And I know I talked about this, I think, with Dr. Herzing when we were out on the boat and discussing, do you number them, do you not, that kind of thing. And I don't think it's necessarily anthropomorphizing. And if it is, it's not a bad way to do it. And it just helps us be able to recognize them and to know them, and also to see them as individuals, right? If you number something, I don't know, it just takes away a little bit, right? So if we're trying to show how amazing these animals are and that they are individuals and they do have lives and they interact with other species and whatnot, and having a name, I think, really helps the public to be able to connect with them as well. And people have asked me like, who gets to name them? Well, mostly me, but and my team. But I kind of have first priority on it. So some of them have random names. Some of them are more have more of a reason for them. But we do try to do the same as I think Jane Goodall is the one who started, where you do the mother and you have the same first letter of the calf, so you can kind of keep the family line. And then if you can, keeping the names together. So like piano has pizzicato, and percussion and piccolo, which are all music names. And then Piper is the granddaughter, or the grand offspring. We don't know the sex of that one.
Well,
That's
for
adorable. It's adorable.
For the A's, we have Annette and Alma, and just like suggestions for some names. Hey, Joe has a question. What do you know about ones that hang out around Shill's Hole?
So we have a few. I don't know much about the ones around Shill's Hole, but we do have a few community scientists who go out to the beach there and take photographs. We haven't made any matches. That's for sure yet, and maybe never. I don't know. Again, we don't know how far they go. But we do know that is another hot spot. So if you're down in that area, Shill's Hole is a really great place to go out. They just go out to the beach and they come close enough that you can see them and perhaps see a mating attempt. They've seen them many times down there.
That's awesome. Oh, Christina just left, but I know she can still hear us. I was curious about because I know where we stayed. I know where the Airbnb, I can tell you how to get to the Airbnb, but I don't know what area that is.
I think that was Susia. Is that how you say it? Susia?
Oh, Susha Island. Mm-hmm.
Okay, because there were quite a few. They were far, but there were quite a few of them.
Yeah, I mean, they're everywhere. I mean, it's depending on where you go, but like a lot of times you go out in the water, you'll see them. Like even if it's just a blip, bloop, and then they're gone. They're all around the San Juan Islands, and in the South Puget Sound and up in the Strait of Georgia. And we have colleagues in Canada that are working on harbor porpoises up there and taking photos. And so we're in the process of seeing if we ever have any matches between those two and that kind of thing.
Is this something that's unique about the San Juan Islands and the Salish Sea? Is that I think it's fascinating that you're able to do this land-based data collection. It's awesome. Do you get out on a boat ever to do your research? And also, is it just something unique to
this area?
Or like your other colleagues around the world, do they also do land-based studies?
So the Golden Gate Bridge, they just literally stand on the bridge, and the porpoises come in during flood tide, come underneath there. And that's another one where they came back to that area. This very similar story as the Salish Sea. They were gone from the bay, and then they've come back. So we like to call them that the Golden Gate is their stationary drone. They just get out above it. So there's the other, I believe the one up in Canada, they have a land-based site. And the one out in Denmark or Netherlands, I think that's on a boat. I don't think that's land-based. So I think there's other locations I think that it can work. The only issue is being close enough to get photos. So for example, Rosario Head, which is down by Deception Pass, is also a great place for sighting harbor porpoises, but there's no low vantage point that we can get out to take pictures. So it's this kind of a Burroughs Pass is kind of a really awesome, maybe unique place. I don't know. I'm sure there's other places somewhere that you could do it from, but I don't know of them specifically. Going out on boats, we actually do have an eDNA study going on. So eDNA is environmental DNA. So you can basically like scoop up sand or water, and then you can pull out the cells of different species and that have been there, and you can get their DNA from there. And so we've started that last year. And so we get out on boats to go do that. So we have to get up, and you have to get up where that fluke print is. So the animal dies, and you have to get to that fluke print and scoop water. And of course, being harbor porpoises, they are more tricky than other animals and other species, that you have to collect twice the amount of water to put through the filters than you do other species, because they just don't shed as many cells I think just because they want to be difficult. Just kidding. But it's very on brand for the species, being shy and evasive, and they don't, love to be in the limelight. So we do get on boats for that, and we just got a new to us boat that was very generously, basically, very cheaply donated to the project. We got a new engine on it, and we're just waiting we're having some trailer issues. So if anybody knows about a trailer, like I have a 23-foot boat I need a new trailer for, please let me know, because that's the issue we're having. But we will be going out more on boats to be able to, we have to get out to different places to find how far we get to have those matches, right? And then to do the eDNA work as well.
Cool. That eDNA sounds fascinating.
Yeah, it's really, we're waiting. We've sent in, like, I think it was 12 or 13 samples from the fall that we should be getting the results back for like any day if we caught any cells, because sometimes it's just luck as to whether there was enough cells in there to be able to get the DNA out. So fingers crossed. But we should be able to then compare with other places, like, oh, we could compare up with Canada, and then eventually we get more. We could compare with South Puget Sound. Are those animals genetically different? That would help answer that community question: are these resident to a particular area, or do they mix with other areas, or is it one big giant Sailor Sea population, which I highly doubt, because they actually have found through eDNA in Alaska, they have those big stocks up there, and they've found genetically different groupings within the identified stock. So there are smaller communities that seem to have natal philopatry. The animals stay there. They're basically resident to that region or area. So I think we're likely going to find a similar thing here, where there's the Sailor Sea population, but we have smaller segments within it. But we have to get the eDNA to find it.
You guys are, I'm looking at back again, like just at this picture, and it's like, to you know what to look for, but that doesn't look like a whole lot of differences, for someone so I'm sure that's taken just, tons of time to learn them, because I see what you're talking about when you talk about the different shadings, but because they don't breach, and because you're doing it from land. You guys are skilled, like that, like really, that's really skillful to be able to identify them, because that's does not seem to be a huge difference, to someone who doesn't do that for a living.
Giles may loan you Eba. We love Eba. Sorry. We love Giles and we love Eba. But yeah, no, the what's interesting is that when we first started, I was like, the dorsal fins are all basically the same. We can't really use those, and unless they have nicks or notches, which is they do sometimes get. We have a few animals with those, which we love because it makes it so easy to identify them. But what was interesting is just within the last year, basically, I was going through the ID catalog, and I was like, I can oftentimes ID individuals just by the dorsal fin, even though they're not that, it's not as distinct as we think for other animals. And it's just because I just look at their photos so often, right? That's really all it is. And I've been doing photo ID for a long time. So I knew the bottlenose and spotted dolphins out in the Bahamas. I knew about 100 of each of those by sight. So it's just been doing it for a while, but it is 100% more difficult than most other cetaceans that we do photo ID for, because they don't always come all the way out of the water. So you don't always have that pigmentation pattern that you're looking at. You're looking at these slight little scar here or there. And that's why we have multiple markings that we use to make sure we're identifying the right animal. But I was surprised at how different the dorsal fins are when you really start looking at them. But
I am just wondering why there's no mention of Dall's porpoise. Are they that rare that you just can't include them in a study, or you just want to focus on harbor porpoise?
Well, it started just so that we knew the harbor porpoises were there, so we started that. And within that, we never saw Dall's porpoises there, and still have never seen Dall's. Huh. The interesting story with harbor porpoises and Dall's is that when the harbor porpoises were around, you saw very few Dall's. Then in the 90s, when harbor porpoises were gone, the dulls came up. So if you have talked to somebody who was out in the water in the 1990s, they're like, oh, if you say porpoise, they're like, yeah, dulls porpoise. But then if you swap back, so in the early 2000s, when harbor porpoises came back, there were very few dulls. And so now people who have been on the water since the early 2000s haven't really seen dulls porpoises. And so, you know, why that happens also, we don't know. They overlap in diet to some degree, but they don't tend to overlap in habitat as much. Harbor porpoises are generally more shallow water, closer to shore, like under 100 meters. And Dall's porpoises generally like a little bit deeper, like 250 plus. But so I don't know why they both can't be here, but they apparently cannot. So you do see Dalls every once in a while, and there's been more recently there's been some sightings that have been a bit more common, but they're just not around. And then particularly in our study site, they're just not there. So I do have some people have asked if I was interested in getting Dall's photographs, and I certainly am. I do have a small little catalog of photos that we have from years past from other people. So if you get Dall's porpoises, I'm more than happy to take them and have a little catalog going for them too. It's just we don't see them very often.
Okay, thanks.
Mm-hmm. Awesome. See, I don't know if there's any other questions. I'm gonna look, but I'm gonna since we talked about Eba, we'll just share.
Eba! Yay! Eba was the best.
We love Eba and her kisses. We love
her. She's got the best kisses. Yeah, and then so with that, so people ask too, like, oh, well, you collect their poop, and I don't even know if we can. I don't know if they poop at the surface or if they poop below. People have talked about, other researchers have talked about glitter water. So sometimes you'll see that they poop because the scales from the fish have come off, and so it's glittery. Mm-hmm. But I don't know. Like, I think of the dolphins. You know, Alma, you're in the water and they poop, and it's like kind of bird poop, and they got these big clumps, and you scoop that up. I don't know if they'd be clumpy enough to do it. I don't know if we could get at the surface. They're so small that she can smell orca poop because it's like really big. So I mean, who knows down the line? But yeah, that one's probably going to be even trickier than the other tricky things about these animals.
When you talk about down the line, I mean, really just hands up to you for doing this work, because we are gonna be able to look back and say, oh wow, when Dr. Cindy started this, and look how far we've come, and look how much more we know. So just thank you so much for your contribution to studying them, because the more we can relate to them, the more we advocate for them. That's just humans, right? And as much as we can share with folks and get them interested in a population, as we've seen, the fight's not easy, but at least there's a relation, because humans don't do a very good job of advocating for nature when it's as a whole. But when you kind of start to introduce them individually or as family units, I think it does bring people's more interest, the more interest, the more education, the more advocacy. And that's what we're all about. So saving these species. So thank you, Dr. Cindy, so much for your work. Really, really appreciate you and all that you're doing. And Alma, thank you so much for inviting Dr. Cindy, because this was fascinating. I took a bunch of notes, and we don't take notes, but I did take some notes and some things I want to look up, and might send you some emails.
And if anyone has any questions, feel free to email, and thank you for that. Like that's what I've been trying to do. I've always been out in the Wild Dolphin Project. I was the bottlenose are cool too, because it was mainly kind of the spotted stole the show, and so it just made sense for me to come here and be like, porpoises are cool too. And so that's been my kind of life's thing, to be like, you know what, let's raise awareness about these little guys and have them be seen as the amazing creatures they are, and that they are your neighbors, right? We talk about our human neighbors, but they are animal neighbors, and they're right in our backyard. And you can go, if you live here in Anacortes, you just pop out to Washington Park and you'll see them if you go out there. So it's really important for us to know about them in that way, and recognize how cool they are. So I appreciate you guys having me out here and on, and getting to share about them, because I just think they're super awesome.
Really great. Thank you. And I just want to encourage people to go to the website and look around. There's a lot of information on here, some cool photos too, but talks about the research, about us, research, how to get involved, news and events, donate. You guys go and donate. This is good stuff. This is good work. And I know I will be doing that. So thank you all so very much for joining. Before we close, I do want to remind folks, and I'm going to ask Deborah here a couple questions. Deborah, what's our deadline? When are you taking these love letters over to the governor's office?
Well, I'm not real sure. Still waiting to see what Peggy has that date. I think it's a little bit fluid depending on if I get an appointment with him.
Okay.
So, they're really adamant about not following up. So I got to do that wait and hear from them kind of thing, which is kind of like, ugh. But do have the basket going. And somewhere I had my first Valentine done. So we got to find Peggy's links. Do your selfies with your Valentines. Keep sharing the word because it just has to happen. It just has to happen.
Thanks, Deborah. And just to remind folks, I don't know that, and the reason I was asking deadlines is I don't know if you go to the post office at this point, please do like a 2-day or something like that to make sure that it gets there. Because if you send regular at this point, it may not get there. So just keep that in mind if you're going to mail something tomorrow. Make sure
I think she was saying like the 13th or 14th to have it to me. So that would mean like Monday, tomorrow, unless you maybe lived down the street.
There's an address there if you guys want to take a screenshot. 1821 Schooner Lane, Oak Harbor, Washington, 98277. And Cindy, if you're wondering what the heck we're talking about, we all made Valentine's for the new governor. Just welcoming him to office and thanking him in advance for his advocacy and support for breaching the dams and saving the southern residents. And so he's getting lots of love letters from folks and some love creations as well. And because it is getting late, and Cindy and others who might want to do something, draw your Valentine. You can also email it to teamcitation@gmail.com. So folks, you can take a screenshot of that or write that down, teamcitation@gmail.com. And what Peggy's going to do is she's going to gather all of them, send them over to Deborah. Deborah's going to print them. Yep, teamcitation@gmail.com.
This is
So
really
the
cool.
deadline is February 14th. It's Valentine's.
Okay, that's what I thought. 13th, 14th. This is from Elizabeth in North Carolina. Look at how cute this is.
Oops, let me take that.
Oh,
There we go.
She's got like their names of the orcas, of individual orcas with little notes. Aren't they so cute, right?
Really cool.
So
awesome.
cute. And then she put a whole typed letter.
wow.
And then I thought this was pretty cool too. Pink power color. She took photos of them out in this, look that's how simple it can be and be impactful at the same time. Pretty
Powerful.
cool. Pretty
Talk
cool,
about
Elizabeth.
science communication and like connecting people and the photos and especially artwork because it shows your connection to your own unique connection to the animals, and I think it's very powerful for humans to see that, and it's great that everybody's doing that and sending it forward.
Yes, I do agree with you, Cindy, that you do need to relate to them as the beings that they are.
Mm-hmm.
It does make us realize that they are part of our family.
That's right.
Rita's
Like
was asking
piano.
for the zip code. The zip code is 98277, Rita's. And there's the letter there. But again, guys, it's not too late. Even if you're not going to get to the mailbox in time, you can go ahead and take some photos and email them to teamcitation@gmail.com so we can get them in there. It's a big push. We should probably do a little post just saying that about the digital, because I think people could do that today, right? And get to you. So we should do something separate with that. Brandy?
And I spend a lot of time at FedEx just because I've dropped stuff off for work. I know here FedEx, so I don't know if maybe if it's a national thing, they are doing specials for the month of February for 2-day for $10. So it's like,
Wow.
that's what I'm going to be doing is running over to FedEx tomorrow and do my 2 days for $10. Yeah. So I don't know if it's only California, but I would think it's everywhere because FedEx usually, whenever they do anything like that, it's across all the states. So you can also
Awesome.
think of that.
No, thank you. That's really, really good to know. Folks, it's not too late. Go do it. Please draw something. Go get some pink paper. My daughter gave me a heart, the plasticky-ish looking paper. Oh my God, I can't even talk. The paper tablecloth. And I used that to, contribute to my art piece, my kindergarten art piece that I'm sending off. So anything at home, and again, even if you just go on there and print a picture or download a picture and do some kind of, there's so many things you could do with your phone now, just take a screenshot of a picture and write on it with pink writing, or something, and just to make it Valentine-y.
Cool.
We can do it. I have Toki's, one of my staff drew I have this giant board that clearly I don't use because I just have a giant drawn Toki on there. And they came in and drew hearts all around her and made a heart hat instead of a salmon hat that she was wearing for Christmas. And so and I thought about just taking a picture of that because it's just, it's creative and it is about love. And this is for Toki's family. So guys, it's not too late. Howard,
and everything that you're doing for the orcas helps everybody else in the Salish Sea, including the harbor porpoises, right? So it's not just one species, it's everybody that you're helping.
all of them. We got to breach those dams.
Yep.
Absolutely. Deb Cruz, you got anything before we close? You good? Howard?
Nope, I'm good. Just scanning the water for whales.
Christina?
Yes, please write your letters to Governor Ferguson. Love letters for orcas. It's not too late. And we need it. We need many. It's just really important. And he's new, so I know he's just going to turn into an orca dork, yep, just like us. So please, yes, 137 species, including the harbor porpoises. So yeah, we got to get those dams breached. Thank you.
Yeah, thank you. Alma?
Thank you, Dr. Cindy, for that fun and fascinating presentation. It was
Thank you
really,
for having me.
really good stuff. And thank you for the good work you do.
Thank you.
Hope to see you soon.
Yes, hopefully, maybe out in Stenella.
Yeah, and maybe us TokiTV people will come and join you on your rock.
Oh yeah, absolutely. Anytime you guys are up here, please reach out. It's just a short 10-minute hike out. That's the one thing that's great about the land base too, is that it's 5 minutes from your house and then 10 minutes out walk and you're out seeing animals. So reach out anytime.
That's really awesome. Thank you. And this group will take you up on that. So
Absolutely. Great.
We'll all be out there on the rock. Brandy, anything last minute besides our FedEx notification?
You know, I did want to ask you about your Canada. Is there anyone in Seachelt or Davis Bay that you have anyone out there? Because my cousin lives out there. So when I go out there, I was wondering about that. Like, do you have anyone taking pictures? Because I know they have a lot of activity out there, but I don't know if I've ever seen anything with porpoises. So I would like to put that on her radar.
Yeah, no, we need more. I think there's a few people, I think maybe 1 or 2 people we have on our community science app that are on the Canadian side, but certainly we need to spread more word up there as well. So please spread away. We need more and more up there. And we can connect with the other organization up there that's doing harbor porpoises. But
Awesome.
if we all put it on our sighting app, then we share it with everybody that needs it. So
Okay,
yeah, it'd be great.
Awesome. Guys, don't forget to go to the website and check out this is the research, but you could just go to pacmam.org and you can see everything that they're up to. Cindy, thank you again. Deborah, thank you for getting that to the governor this week. Deb, please stay on. When we get off, though, I have another question about that delivery offline. And I love you guys. I'm going to close by sharing the live stream from yesterday, just a quick minute, but you guys got to go check out the Whale Museum on YouTube and look at what they did, because look at what they did. What J-Pop did. Because man, did they put on a show for you guys. So we'll close with that. You guys have a great week and we'll see you next week.
Yeah, thank you so much.
Thanks, Cindy.
Thanks, Cindy.